So today we're starting topic 4, movement analysis, and we're going to first begin by looking at neuromuscular function in 4.1. And what that is basically how the message gets from our brain and our nervous system to the muscle and for it to contract. So we're going to have a look at that pathway of how that message travels down our nervous system and arrives at the muscle, and then exactly how the muscle contracts and what's involved in that shortening contraction of the muscle. So we're firstly going to do a quick little recap of some of the anatomy that we should already know. This is the anatomy of skeletal muscle. So the biggest layer, the greatest layer that we've got there is the muscle, of course, and it's surrounded by the epimysium, which is that sheath layer that protects the muscle. At the second layer, we have the fascicle, and this is surrounded by the perimysium, and the fascicle is made up of bundles of muscle fibers, which are surrounded by the endomysium. And today we're actually going to look even deeper than this at the next 3 levels. We've got level number 4, the myofibril. This is an individual muscle fiber cell. So this is just one cell. One myofibril is one cell. And those cells are surrounded by something called the sarcoplasmic reticulum, which we're going to see in the next slide. On the next level deeper, we have a sarcomere, and then underneath that, a sarcomere is made up of myofilaments called actin and myosin, which we'll be talking about a lot today. So let's look at those 3 lower levels a little bit closer. So the myofibril, I said it's a cell of the muscle fiber. It's the contractile element, and it's composed of bundles of myofilaments, which are arranged into sarcomeres. So you can see it's got that striated appearance. It's got all the lines across it, and those lines are individual sarcomeres. And I said before that the myofibril was surrounded in something called the sarcoplasmic reticulum. You can see that in the picture. It's that blue sort of tubular wrapping around each of those individual myofibrils. The sarcomere is going to be, the sarcoplasmic reticulum, is going to be really important because it contains calcium. It's where calcium is held within the muscle, and we're going to understand why that's important a little bit later. So if we look at the sarcomere, you'll see this lined sort of appearance, and it's always arranged into this same sort of arrangement. If you're looking at the sarcomere picture, there's these blue lines and these pink lines, and those are our individual myofilaments. So the thick line, the pink thick one, is the myosin, and it's got these broccoli little heads that are coming off it. And those are the heads of the myosin, which are going to be important for contraction. So it's made up of myosin, which is the thick pink band, and then the blue band is the actin, and these are the thin filaments, we call them. So actin and myosin are our myofilaments. They're arranged into a sarcomere, and there's multiple sarcomeres within a myofibril or a muscle cell. There's an important couple of things on actin that we need to wrap our heads around as well. Firstly, on actin, they have binding sites. Now, we can't see the binding sites here because the binding sites are actually covered by the tropomyosin. So the tropomyosin is that orange band. It's a type of protein. It's just this orange band that runs along the length of the actin, and it blocks the binding site when the muscle is relaxed. So this picture here, we can assume this is a relaxed muscle because we can't see those binding sites. They're covered by the tropomyosin. The other important thing to note on actin is something called troponin. So troponin, it's a little molecule that's attaching the actin with the tropomyosin. It's going to be important, and it's going to come into play when we talk more about what calcium does within the muscle cell. So binding sites, tropomyosin, and troponin are on the actin. Now, I told you that we're going to talk about how the decision to contract a muscle comes from our nervous system to the muscle cells for them to contract. So let's talk a little bit about how that relationship happens. And that happens through a motor unit. It's where the nervous system meets the muscle. So a motor unit is one motor neuron, which is that nerve cell that we can see there in blue. That's the motor neuron, and all of the muscle fibers that it innervates. So some motor neurons attach to heaps of muscle fibers, and some motor neurons attach to only a few. But this right here is our motor unit. So it's one neuron and all of the fibers that it attaches to. And there's a few important structures of the motor unit that we need to be aware of. They're labeled here: the dendrite, the cell body, the nucleus, the axon, the motor end plate, the synapse, and the muscle fiber. So take a second now if you want to pause and try and label those, because I'm going to show the correct labeling right now. So here we have the labeled structure of a motor unit. Firstly, the dendrite is those little prickly bits that come off the top of the motor neuron. The dendrite receives messages from other motor neurons. So think about the nerve cells being in succession with each other. They're all attaching to each other from the brain up until through the spinal cord down to the muscles. There's lots of neurons that connect to each other, and they connect and send messages into the dendrite of the next one. The cell body is where all of our organelles are held, including the nucleus, which is the main sort of structural part of the cell body, and it houses the genetic material of that cell. Then we have the axon, which is like the tail of the motor neuron here. So it's that long part coming down. And the junction between the end of the nerve cell and the muscle, this is called the neuromuscular junction. I don't have it labeled on this picture, but we can see it a little bit close up here. So at the neuromuscular junction, the things that we need to know are the synapse. So the synapse is the area between the end of the neuron and the start of the muscle. So there's a little junction there, there's a little space, and we call that the synaptic cleft or the synapse. It's that space in between the nerve and the muscle cell where that message is going to be passed through for that muscle cell to actually contract. The motor end plate is the beginning of the muscle cell. So you can see there the motor end plate, it's the start of the pink area, it's the start of the muscle. Now, when we contract a muscle, the message is going to get sent in the form of a neurotransmitter called acetylcholine from the nerve through the synaptic cleft to the motor end plate, and that acetylcholine is then going to start the process of muscle contraction. So acetylcholine is going to be important as well. And then we can see, of course, the muscle fibers labeled there in pink. So let's look at how the muscle actually contracts. So firstly, we have the initiation. So the beginning of a muscle contraction starts with a nerve impulse in the motor neuron, and we call this impulse an action potential. It's just that message getting sent through the motor neuron. Transmission, the second step, is as the impulse travels down the neuron's axon and reaches the neuromuscular junction or the synapse at the end of the nerve. So transmission is we send the message down the motor neuron through the axon to our neuromuscular junction. Step number 3 is neurotransmitter release. So what happens then is the nerve cell will release acetylcholine into the synapse, and that acetylcholine will be taken up by the muscle. Now, when that acetylcholine gets into the muscle, what happens is it tells the sarcoplasmic reticulum to release its calcium. All right, so calcium is going to be released into the cell. When we've got acetylcholine present within that neuromuscular junction, when it's taken up by the muscle is going to release calcium in response to that. Now, there's another neurotransmitter called choline esterase, and choline esterase just blocks that message from the acetylcholine, so it stops the release of calcium. So two neurotransmitters that we need to know. Acetylcholine tells the muscle to release calcium, which results in the process of muscle contraction. Choline esterase stops that process of muscle contraction. It reverses the process. It's for relaxation. And then step number 4, the final step, is once there's calcium within the muscle cell, this initiates the sliding filament theory of muscle contraction. So we're going to look at the sliding filament theory of what is actually happening within the muscle for it to contract now. So the sliding filament theory is all about sarcomeres, really. What we want is for those sarcomeres to shorten, for the muscle to shorten. So the sarcomeres are going to shorten. We're going to have millions of these sarcomeres within the cell shortening all at once, creating the contraction of the muscle. So what happens is the thin filaments, the actin, is pulled towards the center of the sarcomere by the heads of the myosin, by the thick filaments. So in some way, the head of the myosin, those little broccoli branches, are going to attach to the actin and drag them into the center of the sarcomere to shorten the sarcomere and to shorten the muscle. And so it's going to come in towards the center like so. Now, we said that when acetylcholine is present within the muscle cell, there's calcium release. So what happens is the calcium is released into the cell. It's housed in that sarcoplasmic reticulum. When it gets the message from the acetylcholine, it's released into the cell, and we can see all those little calcium dots in the picture, all those little red dots coming into the muscle cell. The calcium is going to attach to troponin. So calcium attaches to troponin. When this interaction occurs, once the calcium has attached to the troponin, it's going to cause the tropomyosin to change its shape and reveal the binding sites. So in that part 2 of the big picture there, you can see now the binding sites are visible. We've got those little holes on the actin that we can see. Now that the binding sites are visible, the head of the myosin can attach to the binding sites and can pull the actin in towards the center of the sarcomere. So calcium comes into the cell, attaches to troponin. This interaction causes the tropomyosin to change its shape to reveal the binding sites for the myosin head to attach and pull that actin into the middle. So let's have a look at how that would actually look within the cell. So we've got a video here. We've got the calcium coming in, those little pink dots coming into the cell. It's going to attach to the troponin like so. That is going to make a change in the shape of the tropomyosin molecule. So now the tropomyosin is going to move away and reveal the binding sites so that the head of the myosin can attach like so. So that's the first part of the sliding filament theory. Now, once the head of the myosin is attached, it uses energy from ATP. I know there's a lot going on in this diagram, but I'll just tell you the things that you need to know. Don't worry about the ADP and the P molecule there. It's a little bit deeper physiology. You guys don't need to know that, but just know that the myosin head uses ATP for energy. It cocks its head and releases that energy to pull the actin in towards the center of the sarcomere. Once it pulls it in, it releases. It gets ATP again to hydrolyze, which means to stand back up. So you can see in that fourth picture of our circle, the head of the myosin stands back up and it's ready to attach again to the actin binding sites and pull it in again closer. And this is a circle, so it continues and continues and continues happening so long as there's calcium present within the muscle cell. If there's no more calcium because of the choline esterase, remember that neurotransmitter that blocks the release of calcium, this process stops. So as long as there's calcium present in the cell, we'll just keep going around in a circle like this, and the myosin will keep pulling that actin in towards the middle. So here we go, sliding filament. You can see how it works. So the heads of the myosin attach to the blue actin and continue to pull it in so long as there's calcium within the cell. Now, when we look at a sarcomere, there's a few important bands or zones within a sarcomere that you guys have to know. So let's start with the H zone. Now, the H zone is the central region where there's only myosin present. So you can see in the first picture, in the contracted muscle, the H zone is quite large because the actin is out to the sides, versus in the second picture, the H zone has gotten smaller. It's only in that middle part where there's only myosin. The A band is the entire length of the myosin filaments, and it remains consistent during contraction. So you can see that the A band hasn't changed from picture 1 to picture 2. It's the entire length of the myosin, which doesn't change. The Z line is like the boundary of each of the sarcomeres. It's where the actin is connected to. So when that muscle contracts and the actin is pulled towards the center, the Z line comes in towards the center as well. So the H zone, the A band, and the Z line, those are the only ones I want you guys to know about or to highlight there. All right, so let's do a little recap on that. Number 1, the motor neuron produces the action potential, which is just the message that travels down the axon to the neuromuscular junction. At the neuromuscular junction, acetylcholine is released into the muscle cell, which causes the release of calcium from the sarcoplasmic reticulum. Calcium floods into the cell and it binds to troponin, which changes the shape of the tropomyosin and moves it away from the binding sites on the actin filament. Now the myosin head can attach to the actin and pull those actin filaments in towards the center of the sarcomere using their energy from ATP. All right, so just a quick bit of information here as well is that there's a few things that influence how a motor unit works. So remember, a motor unit is one motor neuron or one nerve cell and all of the muscle fibers that it innervates. Firstly is the size of the motor unit. So I said some neurons innervate lots of muscle fibers, some innervate only a few. So we're going to find big motor units in our big powerful muscles like our quadriceps, and we're going to find small motor units in our hands and feet where we need to create precise movements. So our big motor units that innervate lots of muscle fibers are going to produce power, versus where we have more small motor units, it's going to be more for precision. Secondly is the stimulation frequency. So how much of a message is the nerve sending to the muscle? Is the nerve saying, contract really, really, really hard? This is like a maximal effort. You're working really hard in the gym. Or is it just like a reflex where we have to quickly pull away? Is it a short little muscle contraction? So in the first part of our picture here, we've got one stimuli. So this would be something like a reflex versus a lot of stimuli, which would be something like we see in the gym where that nerve is just consistently sending the message to the muscle to contract, and we're able to produce a more forceful muscle contraction. The second thing is muscle fiber type. So there's a few different types of muscle fibers, which I'm sure you've heard of before. If we have a look at our muscle fiber again, when we looked at the anatomy of the skeletal muscle, so the muscle fiber, number 3 there, it's easy to think that they're all one and the same, but as you guys know, there's actually 3 different types of muscle fibers, which are called type 1, type 2A, and type 2B. Now, typically, people remember these by different sports and which muscle fibers are most useful for which type of sports. And we all have a different ratio of type 1, type 2A, type 2B muscle fibers. Generally, it's down to genetics, but it also can be due to training. With certain types of training, we can actually change the type of muscle fiber that we have. So type 2A can transform into type 2B over time with the right training. So type 1, it's our endurance muscle fiber. Generally, like our marathon runners would want to have a high ratio of type 1 muscle fibers. Type 2A is a more powerful muscle fiber, but it still can sustain that power over a medium sort of set of time. So that picture there, think like 400 to 800 meter running. That would be type 2A muscle fibers doing a lot of the work there. Versus our type 2B muscle fibers, which are our super powerful muscle fibers. They produce really strong contractions, but they fatigue really quickly. So you think about your 100-meter sprinter or your weightlifter who's able to produce a very forceful contraction, but you can't continue doing that again and again. Those muscle fibers are going to get very fatigued. So in terms of telling the differences between these three types of muscle fibers, they can be differentiated by two things. Firstly, energy. How is the ATP actually formed within these muscle fibers? And secondly, is the speed of contraction. So how quickly are they going to contract? If we look a little closer at our type 1 muscle fibers, their speed is slow twitch. Their energy is aerobic metabolism. So they're getting their energy from oxygen, from aerobic metabolism. And if we put the speed and the energy together, we get the classification, which is slow oxidative. So they produce slow speed contractions and they use oxygen for their energy source. Their force is generally low and their fatigue is low as well. So even though they're not producing high forces, they're able to continue contracting over long periods of time. Because they use that oxygen as their energy, they're able to keep producing energy over time. So generally a marathon runner, cross-country skier, any long sports are going to use type 1 fibers. Also, something to think about as well is our postural muscles. So if we're standing up for a long period of time, we want those muscles in our back, which are going to help us with our posture, help us stand up, to be contracting. So the postural muscles are called the erector spinae. They're like big columns of muscle that run down your spinal cord. And we want them to be able to not fatigue very quickly. We need to use them for a long time. So our postural muscles generally have quite a high volume of type 1 muscle fibers as well. Type 2A, these are fast twitch, so think more quick, powerful movements. They get their energy from aerobic metabolism and some anaerobic metabolism as well. So not only are they using oxygen for energy, but they are also using glycogen through the anaerobic pathways too. So type 2A is like right in the middle of type 1 and type 2B. The classification is fast oxidative. Their force is high, so they're producing high force movements, and their fatigue is intermediate. So we're thinking sports of between like 20 seconds to 10 minutes is going to be important for our type 2 B muscle fibers, where we want to produce high force. We need to sustain that over some period of time, but not a very long period of time. And then finally, our type 2B, these are our fast twitch muscle fibers. They get their energy from anaerobic glycolysis, so they're using glycogen as their energy. Their classification is fast glycolytic. They're able to produce very high force contractions, but they fatigue very quickly as well. So high fatigue rate, you're not able to do this. You're not able to produce this force for a long amount of time. So there's the classification of our muscle fibers all together. And something interesting about muscle fibers as well is they actually have different colors. So type 1 muscle fibers are oxygen muscle fibers. They're using oxygen for energy. Because there's a lot of oxygen and a lot of mitochondria within that type 1 muscle fiber, they are red in color. Our type 2A are pink in color because they're that halfway point between oxygen and glycogen. And type 2B, where there's no oxygen helping to create energy within that cell, they appear white in color. Now, when we have a look at the sort of the marathon runner versus the sprinter, there's a few things that we notice immediately, the differences between these two athletes here. Probably the most obvious thing is that the sprinter is much more muscular, is much bigger. The marathon runner is typically a lot lighter. But something that you might not notice about the difference between these two athletes is their age. So the average age of a successful marathon runner is 38 years old, whereas the average age of a successful sprinter is 26 years old. And there might be many reasons for this, but generally when you look at endurance sports, the athletes are quite a bit older than power sprint type sports. And one potential reason for this is because as we get older, our type 2 muscle fibers actually decrease. So you can see in this graph here along the y-axis, we have the area of the muscle fiber, so the size of the muscle fiber. And along the x-axis, we have the age. Now, if we follow that line that's going straight across, that's our type 1 muscle fiber. So with age, there's no change in the size of the type 1 muscle fibers. However, with age in type 2, you can see that line is sloped downwards. So as we get older, our type 2 muscle fibers actually decrease in size, which leads us to the conclusion that power declines at a faster rate than endurance in terms of the muscle. All right, guys, that's the end of 4.1.